Good day everyone, my name is Senator Idris Erinlakatabu. The topic we are to look into today is a cellular respiration. When we're talking about the cellular respiration, you can simply say cellular respiration is a series of enzyme control reactions in which oxygen is involved in breaking down of a glucose into carbon dioxide, water, and a chemical bond energy called ATP. Cellular respiration usually occurs in the cell and specifically in the mitochondria of the cell. The cellular respiration can be symbolically represented by the following equation, where we have the glucose as 6,6H12O6 plus the oxygen we're talking about, that is 6O2, and it's going to result to 6CO2 plus 6H2O plus ATP. That is the energy we're talking about. That means this stands for glucose. This stands for oxygen. Then this stands for carbon dioxide. Carbon dioxide, just say carbon dioxide. Then we have this as water, while we have this as the energy. That is the main product. Now we can simply say carbon dioxide and water, the two of these are byproducts. They are byproducts. That is unwanted materials. While energy here is the main product. The main product, that is the essence of having a cellular respiration, because cellular respiration aims at releasing of this and generating energy. Now, we can see a case where oxygen may not involve in the breaking down of the glucose. And for this case, we can simply say there are two types of cellular respiration now. Let's look at types of cellular respiration. This is based on whether oxygen is involved or not. If oxygen is involved, such type of cellular respiration or respiration is called aerobic respiration. We call this one aerobic respiration. But when oxygen does not involve or is not involved, we call it anaerobic respiration. That is the two types of respiration we have. One, both of them aims at generating energy, but the other products will be different. Now, to look at this one after the other, let's firstly consider aerobic respiration now. Aerobic respiration. Let's consider aerobic respiration. On aerobic respiration, as the name implies, aerobic respiration is a type of respiration that basically involves the oxygen. That is, oxygen is involved in breaking down of a glucose to release the energy. Likewise, we have carbon dioxide and the water. So this cellular respiration equation is meant for aerobic respiration now, where we just say the glucose will be what? That is when we say 6,6H12O6 plus 6O2 is going to give us what? 6CO2 plus 6H2O plus at times we say 38 ATP. That is when what? When oxygen is involved. So we can see that is when oxygen is involved. Now, before we go to anaerobic respiration, let's give further explanation on aerobic respiration. Now for the aerobic respiration, as you can see the equation, this is the glucose, this is the oxygen we are talking about. We have the carbon dioxide as byproduct, water is byproduct, where there is a release of about 38 net of ATP as the main product. Now let's look at the stages of aerobic respiration. Stages of aerobic respiration. We want to look at stages of aerobic respiration. As you can see this, we're talking about the stages of aerobic respiration because the reason being that before we have the formation of the carbon dioxide, water, and this 38 net of ATP, there are stages. There are stages in which these products are being formed. Now, there are three stages of aerobic respiration, and the stages are this. Number one, we can simply look at the glycolysis. Glycolysis as the first stage. That is number one. Number two of it is what we call kreb cycle. Kreb cycle. That is second stage of aerobic respiration. That is the kind of respiration that involves oxygen. Number three of it, we have electron transport system. Electron transport system is what we call, that is the second stage of it, where we that simply called ETS. So these are the three stages of aerobic respiration. The first one is glycolysis. We have Krebs cycle and electron transport system. Let's now look at teach one after the other. We are going to glycolysis now. Let's look at the glycolysis. You see, on glycolysis, as the name even implies, glycolysis, here we mean glucose. Lysis here simply means breakdown. So if something is being split or breakdown, that is what we normally use the lysis for. Now, to look at glycolysis, you can say, as we all know, it is the first stage of cellular respiration. Glycolysis is a series of enzymes. It's a series of enzyme controlled reaction which takes place in the cytoplasm. One. Then we can say it involves the breakdown of 6 carbon sugar. You know, glucose is 6 carbon sugar, that is extra sugar. 6 carbon sugar into two molecules of pyruvates. Usually in form of pyruvic acid. So that is definition for glycolysis. We said it's a series of enzyme control reaction. As you all know, anything that has to do with the metabolic process, so it's involved of enzyme control reaction, and it takes place in the cytoplasm of the cell. So when we say it takes place in the cytoplasm of the cell, it simply means it does not depend or require oxygen. So it is a kind of process that occurs in the absence or presence of oxygen, meaning that it does not depend on oxygen at all. Whether oxygen is present, glycolysis takes place. Whether oxygen is not there, although it does not involve oxygen. So that means glycolysis occurs anaerobically, that there is no oxygen requirement. Now, when I said it involves the breakdown of the 6 carbon sugar into 2 molecules of pyruvates, usually in form of pyruvic acid. So with this now, we can simply represent this with equation. When we say glucose still remains chemical formula, where we have C6H12O6 plus, it's supposed to be plus oxygen, but there is no oxygen. It means is equals to, so what it returns to what? To the pyruvate, and it is 2 molecules of pyruvate. We say 2CH3COCOOH plus 2NAD plus 4ATP. That is the amount of the energy that we generated. So we refer to this one as our glucose. This is glucose, This is pyruvate, or we just say pyruvic acid. This is pyruvic acid, while this one is electron carrier, or call it hydrogen acetone, and this is net of ATP that is generated. That is energy. The ATP is what? Is a standard unit in which the energy produced in cellular respiration is being stored, and that is adenosine triphosphate. So this is the chemical formula for glycolysis. Now, that is the end product of glycolysis. Now we can see the pyruvate has formed. Likewise, we can see certain amount of energy being generated. And as you are looking at this equation, how glucose is being broken down to form the pyruvate and the what and this net of ATP, it's not just something that just happens suddenly like that. It's of systematic process. It's of process that take place, let's say, stepwisely, meaning that it has stage. There are stages of glycolytic process. Let's now look at the stages now. We can say stages of glycolytic process. The first stage is what we call addition of phosphate group. Addition of phosphates. Addition of phosphate group. That is the first stage or first step. 2, we have lysis. Lysis is the second one, while the third one is dehydrogenation. Dehydrogenation. So these are 3 stages or steps of what? Of the glycolytic process. Now we are now going to look at this with the help of illustration. How addition of phosphate group occurs to the glucose and what it later forms, the lysis and dehydrogenation. That is the next step now that we are going to look into. That means we are going to have an illustration of glycolysis now. As the process of glycolysis now in illustration, we say, glucose is 6 carbon sugar. We can write it as 6 carbon sugar. Now, the first thing is, as the glucose is coming down like this, here we have ATP, that is adenosine triphosphate. The adenosine triphosphate will supply P, that is a phosphate group, and when phosphate group is added to the glucose, we are going to have glucose 6 phosphate. We have glucose 6 phosphate. That is the addition of the phosphate. That is the first step. And this reaction is controlled by enzyme exokinase. It is controlled by enzyme exokinase. That is now glucose 6 phosphate will now undergo lysis. I said lysis has to do with the breaking down of a thing. Now, we are going to have the 3 molecule of 6 carbon sugar, which is triose sugar. You can just call it triose sugar. Are we getting it now? Or we refer to it as glyceraldehyde 3-phosphate. That is another name given to it. That is glyceraldehyde 3-phosphate or phosphoglyceraldehyde. Are we getting it now? No, and that 3 triose sugar is 3 carbon sugar. As this one is 6 carbon sugar, this one is what? This one is 3 carbon sugar. The next thing now is the hydrogenation. When we talk about the dehydrogenation, as the D simply means removal. That is removal of hydrogen. So there will be removal of hydrogen now from, let's rub this. There will be removal of hydrogen now. From where? From a triose sugar. And what will now remove the hydrogen? There is something called hydrogen acetone. We call it hydrogen carrier. Or at times we refer to it as electron carrier. So it is this thing that we call NAD. NAD. And what is full meaning of NAD? That is nicotinamide adenine. We have nicotinamide adenine dinucleotide. That is the full meaning of NAD. It is usually called electron carrier or hydrogen acetone. Now, from the triose sugar now, there will be removal of 2 atoms of hydrogen. And when it removes 2 atoms of hydrogen, this hydrogen acetone will now reduce to NADH2, if at all we call it 2 atoms of hydrogen. And from here now, we are now going to have what? 2. For the removal of this now, there will be what? There will be addition of there will be production of ATP. Certain rate of ATP will be generated. And finally, we are going to have the pyruvate that will form. And what is that pyruvate? So this we are going to have pyruvate in form of pyruvic acid that will be written as CH3COCOOH. That is the end of a what? Of a glycolysis. But what now happens next to the pyruvate will determine whether it should proceed into Krebs cycle or it should continue anaerobically or it should continue its anaerobic condition. We can see that this process of glycolysis is anaerobic condition. The reason being that it does not involve any oxygen. That is why glycolysis naturally occurs in cytoplasm of a cell. Now, after the pyruvate has now fully formed, what now happens next will determine whether the oxygen is present or not. If, let's now consider the case now. If at all there is no oxygen, the pyruvate will proceed into another thing, will form another thing, or let's say it continues anaerobically. But if oxygen is present now, that is when the pyruvate will now go to the second stage of aerobic respiration, which is Krebs cycle. Let's now consider the two factors now. The first factor is that if no oxygen present, what is the fate of pyruvate? Let's now see. We say pyruvate reduces into lactic acid. As in what? As in muscle cell. As in, let's say, as in animal cell or muscle cell. Are we getting it now? So that means if there is no oxygen now, the pyruvate will now proceed anaerobically, that is going to reduce into lactic acid. Let's now look at the question now. You can see that this is the pyruvate where we have CH3COCOOH. That is our pyruvate. And once there is no oxygen in the muscle cell of an animal, so what happens is that the pyruvate will just reduce to lactic acid. And this lactic acid can be symbolically represented by CH3COOH 2NAD plus 2ATP, because certain amount of energy will still be generated. So this is anaerobic condition in muscle cell, and this will be the result. The pyruvate will just reduce to the lactic acid once there is no oxygen present, and that is in animal cell. Let's now look at it if it happens in plant cell. We now say pyruvate reduces to or into ethanol as in what? As in plant cell, or in some fungi such as yeast that is commonly known as Saccharomyces cerevisiae. Now, what will now be the equation now? What will be the anaerobic condition of it? Let's give the equation. The equation will be CH3 as ethanol, COCOOH, as pyruvate. This is the pyruvate. Pyruvate will now reduce to ethanol of C2H5OH plus 2NAD plus 2ATP. So that's what we're going to have. We can simply have 2 here as well. So this one will serve as our pyruvate. This is the pyruvate. This is the ethanol. This is what we call ethanol. And why this one is energy. This is still remaining energy. So that is anaerobic condition in plants and animal cell. In animal cell, the lactic acid will be formed. In animal cell, the lactic acid will be formed. In plant cell, the ethanol will be formed. And this formation of ethanol in the yeast is what we call fermentation. We call it fermentation. That is fermentation in the yeast that is result to the pyruvate will reduce to what? Will reduce to ethanol. Some people believe that it is the glucose that will reduce to the ethanol. No. The glucose will first pass through the stage of glycolysis and that is when we are going to have the pyruvate. After the pyruvate has formed, it is this pyruvate we now reduce to the ethanol if at all there is no oxygen. It reduces to ethanol in plant cell if there is no oxygen, while it reduces to lactic acid in animal cell if there is no oxygen. Let's now assume that oxygen now present. For the case of our animal cell now, let's consider that the oxygen now later present. Such an oxygen that later present, this oxygen will have to reconvert lactic acid back to the pyruvate, because it is not the lactic acid or ethanol will proceed into the Krebs cycle. No, it is the pyruvate that must also proceed into the Krebs cycle as the second stage. So as a result of this, we now have a kind of oxygen that will reconvert lactic acid back to the pyruvate, and such oxygen is what we call oxygen debt, a kind of oxygen that is required that ensures the conversion of lactic acid into the pyruvate. Now, let's now consider the second factor, which says if oxygen present, what is the hope or what is the fate of the of pyruvate? Let's look at it now. We say if oxygen present, we say pyruvates proceeds into the second stage of aerobic respiration, which is Krebs cycle. It proceeds into Krebs cycle as second stage of aerobic respiration. Let's now look at the second stage now, which is Krebs cycle now. Let's look at the Krebs cycle as the second stage. To look at the Krebs cycle, once this process or reaction involve oxygen, that means that all activities of Krebs cycle will take place in mitochondria. The activity or process or reaction of the glycolysis take place in cytoplasm because it does not involve oxygen. But in the case of Krebs cycle, it is because oxygen is present. We now have this kind of situation or reaction that it now takes place in the mitochondria of the cell. Now to now look at the Krebs cycle, we say Krebs cycle is the second stage of aerobic respiration, in which the pyruvate that is formed as a result of glycolysis finally oxidized to produce carbon dioxide, water, and certain amount of energy. But before we now go to that, before the pyruvate enter into Krebs cycle, pyruvate has to undergo a kind of a chemical reaction, because it doesn't just enter into Krebs cycle like that. There are certain things that will happen to the pyruvate before getting into the Krebs cycle. And what is that? Let's now have this as a kind of illustration to better explain the process of Krebs cycle. Let's have the illustration of the Krebs cycle now. This is let's now look at the illustration now. We have the pyruvate. So as we have the pyruvate as the end product of glycolysis, I said earlier that before it enters into Krebs cycle, so there will be a removal of atom of hydrogen by NAD, and this one will reduce to NADH. Then there is also a removal of the carbon in form of carbon dioxide, and here we are going to have a CO2 will also be removed. And we shouldn't forget that the pyruvate is 3 carbon acid. And once a particular carbon is lost, it's going to remain 2 carbon. And these 2 carbon, we call it 2 carbon acetic acid. And it's the one that will now react with enzyme-CoA to form 2 carbon acetic-CoA. That is what is going to form. It is these 2 carbon acetic-CoA that will now enter into what? Into Krebs cycle. So here we now have the journey of Krebs cycle now begin. But in some cases, this enzyme-CoA will be lost, so that for another reaction to take place. So that means if the CoA is being lost back to this side, so it means we are going to have acetic acid that will now form. Just a 2 carbon acetic acid. Now, as 2 carbon acetic acid just entered, let's say this is acetic acid now, and we all know it is 2 what? It is 2 carbon. It's just 2 carbon. When they enter into the Krebs cycle, let's say this is the Krebs cycle now. So, from the previous reaction, there is 4-carbon oxaloacetic acid that this two carbon acetic acid will react to form citric acid. That is why in some cases we refer to Krebs cycle as citric acid cycle, or we call it tricarboxylic acid cycle, because there is going to be production of 3 molecules of carbon in form of carbon dioxide. So let's now look at it now by saying from the previous cycle we have 4-carbon oxaloacetic acid, and we should know that 4-carbon oxaloacetic acid is what? This is 4 carbon. It is 4 carbon. So as this one just reacts with this now, so when it reacts with acetic acid, there will be formation of what? There will be formation of, this one is just, let's put it like this one is coming and this one mixed with it. Now we are going to have this, that is a citric acid, and citric acid will be how many carbon acid? This is 2, this is 4, so citric acid will be 6 carbon acid. 1, 2, 3, 4, 5, 6. So it's going to be 6 carbon acid. So as we have the citric acid now, this citric acid will now reduce to, we undergo a kind of analysis and reduce to what we call isocitrates. The enzyme that we manage that is isocitrates. We call that enzyme isocitrates. Now from the isocitrates now, there will be removal of atom of hydrogen by this our common hydrogen acceptor. At the same time, there will be removal of of carbon in form of carbon dioxide. So here we are going to have our NAD that always serve our hydrogen acceptor, and this will reduce to NADH. Then we also have the removal of CO2. So that is a molecule of that, the molecule of carbon. Now after removal of these two, what are we going to have? So it's going to remain another carbon acid that has been removed is going to remain 5-carbon ketoglutaric acid. We are going to have ketoglutaric acid. So and ketoglutaric acid is how many carbon? We are going to have a 5 carbon, 1, 2, 3, 4, 5. That is 5 carbon acid. Are we getting it now? Now, after having this, at the same time, what happens to this isocitrate? The same thing applicable to ketoglutaric acid. So from ketoglutaric acid, we still have a removal of removal of atom of hydrogen by the same NAD, and we are going to have NADH. At the same time, there is removal of carbon. That's the carbon in form of carbon dioxide. The enzyme controlling this is what we call carboxylase, while enzyme controlling this is what we call hydrogenase. So what are we going to have now? The result will be what? The result is succinate. So here there is enzyme CoA. The enzyme CoA will now react. Let's assume we have the enzyme CoA. So the enzyme CoA will react with this succinate and become succinate-CoA. It becomes succinate-CoA. So when it becomes succinate-CoA, there will be phosphorylation of ATP. That means certain amount of ATP will be generated. And that is the condition is where we have the ADP plus P, which is going to give us what? ATP. And that is that. And after the ATP has formed, the final thing that will form is what? Is succinic. We are going to have a succinic acid. And we shouldn't forget that succinic acid is how many carbon acid? Though a particular acid has been lost, a particular carbon has lost. So we are going to have a 5 carbon acid. The 4 carbon acid. That is 4 carbon acid. So we are going to have a 4 carbon acid. Now, you can see the succinic acid as 4 carbon acid. So we also have a specialized kind of hydrogen acceptor. The first hydrogen acceptor that is common is NAD. But we also have a particular hydrogen acceptor that is called FAD. Why the formula of FAD is flaphine adenine dinucleotide. Flaphine adenine dinucleotide. So it is this flaphine adenine dinucleotide now that will remove what? Let's put it here. It is this that will remove this FAD now. Will remove what? Will remove hydrogen from this succinic acid to form FADH. And as the atom of hydrogen has been removed, so the final product will now be fumaric acid. We call it fumaric acid, or we call it fumarate. that is fumaric acid now, or we call it fumarate. As you can see the fumarate now, the fumarate still remains how many carbon acid? Is still 4 carbon because there is no carbon that is being removed there. So we still have this one as what? As 4 carbon acid. Now we now have the fumaric acid. We just reduce this to what? We just reduce this to malic acid, or we call it malice. And malice is still how many carbon acid? That is 4 carbon acid. And from the malic acid, there is a remover. There is a remover of atom of hydrogen, and what remove that? That is a what? That is NAD. We remove the hydrogen and become NADH. So to now form oxaloacetic acid, which is still remain 4 carbon acid. So it's this 4 carbon acid that will now what? That will also proceed the reaction. So this is the what we call a Krebs cycle. As this continues the reaction, this oxaloacetic acid reacts with acetic acid again to form citric acid cycle, and to form citric acid, the citric acid will reduce to the isocitrate. Isocitrate, from the isocitrate, there will be removal of atom of hydrogen by NAD, and there will be removal of carbon in the form of carbon dioxide. That is with the aid of enzyme hydrogenase to form 5 carbon acid ketoglutaric acid. After the ketoglutaric acid has now formed, there's also a removal of atom of hydrogen from ketoglutaric acid to also form NAD, then as well as a molecule of carbon in the form of carbon dioxide. So to now reduce to succinate. So succinate will react with enzyme CoA, and in this process of reaction, there will be a formation of ATP that we call phosphorylation of our ATP, because this is one of the conditions by which phosphorylation of ATP takes place, where there is separation of coenzymes, or the separation of electron and the proton. Now in this case now, after the ATP has reacted with a phosphate group, there's going to be a production of our ATP. Now we now have what we call succinic acid. From the succinic acid as well, so that's the 4 carbon acid. Succinic acid will also reduce to what? Will reduce to fumaric acid as a result of removal of hydrogen atom. So after hydrogen atom has also been removed from this, fumaric acid will form. Then the fumaric acid will also reduce to what we call malic acid, which is still remain 4 carbon acid. So from the malic acid now, we are going to have a removal of atom of hydrogen as well by what? By nicotinamide adenine nucleotide, that is NAD. So to also give us oxaloacetic acid. That is how the cycle will continue. So this cycle now is what we call Krebs cycle. We call it Krebs cycle. Now, for every molecule of pyruvate now that enter into mitochondria, what will be the product? What will be the product? The product is, we are going to have, let's count our product now. You know, how many carbon acid do we have? I mean, let's say how many carbon dioxide do we have? Or let's say how many carbon in form of carbon dioxide, how many ATP in form of our energy, then how many NAD and FAD that are generated. So let's now count with this now. So for the carbon, here we have 1 carbon. This is another carbon making 2. This is another carbon making 3. That's why we call this one 3 carbon. So 3 carbon will form. So from this 3 carbon that generates, that's why we call another name for Krebs cycle is tricarboxylic acid cycle. That is for carbon. For the ATP, how many net of ATP is generated? Here we have only 2 net of ATP. We say 2 net of ATP. That is a time we find that's 1 ATP. Now for the NADH and FADH, how many do we have? Let's count. This is 1. It's like here we have 1, 2, 3, 4. So here we have 4 NADH and 1 FADH. So these are the products these are the final products in Krebs cycle, and that is the second stage of aerobic respiration. Now the last one is what? Is electron transport system. To briefly analyze the electron transport system now, Electron transport system is the third stage. It also takes place in the mitochondria of the cell. As this one occurs in the mitochondria, the other one also occurs in mitochondria. So the electron transport system is one that aims at generating ATP. It aims at generating large amount of energy. And when we go back to the equation and say there is about 38 net of ATP generated. Yes. How do we have this 38 net of ATP that generates? When we look at the glycolysis, in glycolysis we have only 2 net of ATP that are generated. ATP are generated. That is 2 net of ATP in glycolysis. In mitochondria here, we have 2 ATP also generated. But in electron transport system, that's where we have up to 34 ATP generated. The total will now be 38 ATP. Now, before we finalize on the ATP, on the electron transport system, it is the ETS we're talking about, the electron transport system. It's one that aims at transferring of the electron from one hydrogen acceptor, hydrogen carrier, from one hydrogen carrier to another one until ultimately they are accepted by the oxygen. Until ultimately this electron are accepted by oxygen to now form water. So that means we have NADH will be transferred to, let's say this is another NAD, this is another NAD, this is another NAD. Because the oxygen present is one that now lasts this. The oxygen is now present here. That's where we have the oxygen. So the electron will be transferring from this NAD to this. As the electron is transferred from one place to another, there will be formation of ATP. So the more it transfers, the more we get more amount of energy that will be generated. So the ATP, the hydrogen will be transferred here, and this one will become NADH. It will transfer here, and this one will also become NADH. It also will transfer here, and this one also become what? NADH. Until ultimately the hydrogen now accepts this to now form H2O. So to now form water. So that means the water in the equation of photosynthesis is generated in the what? In the ETS. So the two products that we're going to have in ETS are large amount of energy, like about 34 ATP, and we also have water. So when we now combine everything together, we now have the summary of the equation that says, we now have the summary of the equation which come up with C6H12O6 plus 6O2 will result to CCO2 plus 6H2O plus 38 ATP. So that is the equation for cellular respiration. And then you can see the amount of the energy formed in the pyruvate, in the glycolysis, the amount of the energy formed and the products in the kreb cycle, likewise in the ETS. Now this is the end of our cellular respiration. If you have any question, you can ask in the comment section. Thanks. I still remain my name, Senator Idris Erinlakatabu. But don't forget to subscribe to Mr. Pure Biology. Thanks.